I'll talk today about incorporating ecology and evolution into models, specifically gene-drive models. And the biological problem I want to talk about today is that we have some species that are out of control. So we have disease vectors causing malaria, we have invasive species, we have agricultural pests. And we've been trying to control these populations and suppress these populations, eradicate these populations for quite some time now, using things like pesticides and biological controls. But globally, we don't seem to have a solution to this problem. And one idea that has been suggested for quite some time now is to use genetic control. So the idea is you take a deleterious allele and you... Slides are not being shared. Share. Yeah. Yeah. Works?
Yes.
Okay. Right. So we've been trying to control this population for quite some time, and people have suggested this idea of let's take a deleterious allele and spread it in a wild population, and in that way we can suppress the population, right? The problem is, of course, how do you take a deleterious allele and have it spread in a wild population against natural selection? And one solution that has been suggested for this problem is to use something called a gene drive. So what are gene drives? Under normal Mendelian inheritance, we don't expect, if we introduce an allele to the population, we don't expect to see a change in the frequency just because of inheritance patterns. But if this allele somehow violates Mendel's laws and propagates at frequencies or probabilities higher than 50% to offspring, we have something called a super Mendelian inheritance. So in this example, all the offspring of this mixed couple are going to carry the allele. And what we get is an exponential increase in the frequency of the allele, even if this allele is deleterious, right? So now we have a sort of a mechanism. All we need to do to spread our deleterious allele into this pest population is have it violate Mendelian inheritance. And one way in which we can do that is using CRISPR-based gene drives. So here the idea is you take the genetic code for the CRISPR mechanism, and you have this cargo gene, this deleterious allele that you want to spread in the population. And what happens is you inherit, let's say, this from one of your parents, and from your other parent, you inherit the wild-type chromosome. The CRISPR element is going to cut the wild-type chromosome, and then DNA repair mechanisms are going to copy the entire thing into the other chromosome. So basically what we have is a conversion of a heterozygote to a homozygote carrying the gene drive allele. So this violates Mendelian inheritance and allows us to take a highly deleterious engineered allele, and we can connect to it basically any cargo gene that we want to spread in the population and have it spread to very high frequencies. This is a new technology that people are very excited about. But how do we model it? So we can think of a very simple model for this system. We need to think about the wild type, and we need to think about the gene drive homozygote, and there's some fitness costs for carrying the gene drive, right? Where we want to suppress a population. We need to think about the dominance, of course, of the gene drive allele versus the wild type. This is a standard. But what we can do is we can add just one more parameter to this model, which is the conversion probability. So what's the probability that a heterozygote will be converted to a gene drive homozygote? So a perfect gene drive will be C equals 1, but we can think about gene drives with different parameters. So already with a very simple extension of existing models, we can write down equations that describe the behavior of the gene drives. So how does this look like? We need to write down a recursive equation describing the change in the frequency of the gene drive allele from one population to the next. We need to think about the contribution of the different genotypes to the next generation. And the only element that's new here is we need to separate the case for the heterozygotes that remain heterozygotes and for the heterozygotes that are converted to homozygotes and maybe experiencing different selection coefficient. So people can just take this simple model and run it with some parameters. And what we see is that for a wide range of parameters, we get very fast increase in the frequency of the gene drive from very low frequency to fixation in something of the order of 10 to 20 generation for a wide range of parameters. And indeed, when people did experiments, so they took cages with mosquitoes and introduced the gene drive. You can see these tracking the top one is tracking the allele frequency in the cage. The bottom one is tracking the population size of the cage. And what you see is you got a very rapid increase in frequency, about between 9 and 11 generations, I think, is the time that fixation took in this experiment. And you see the populations are all collapsing, right? So the cages were empty after the experiment ended. So it works in the lab. But what we can see is that we have here an evolutionary process that's occurring very, very fast at the same time scale as the ecological process of interest, right? We're interested in this demographic crash of the population. It's occurring in the same time scale as the expansion or the increase in frequency of the gene drive. So in many cases, we can sort of separate the timescales of ecology and evolution, but clearly in this case, we cannot do that. We have to think about ecology and evolution together in gene drive models. So given this perspective, what we did is we sort of sketched out the possible ways ecology can affect gene drive dynamics and sort of suggest this as a framework for a modeling project, right? We need to model all these things in order to understand how gene drives are going to behave in nature when they are released into a field trial or a wild population. And thinking about these, the most important thing to start modeling is actually population structure. And the reason is that this technology is very risky. This is one of the reasons that it's not yet been applied in a field trial. And the risk here is that this element is supposed to spread very rapidly in the population, but what if we wanted to spread only in a single population, right? There's a high risk that this is going to spread out of our population, spill over to other populations, maybe even other species, right? So this is a big concern, so we need to model it to understand what's going on. And the simplest model that we can think about is just a two-population model. We have a target population where we want the gene drives to work. We have a non-target population where we don't want the gene drives to work, but these are connected by some migration. So we can think about, for example, this project called Predator-Free New Zealand. It's a project by the New Zealand government for eradicating, for example, the Australian possum, which is a horrible invasive species in New Zealand, but it's native to Australia, right? So if New Zealand introduces the gene drive to New Zealand, we expect spread of the gene drive very, very rapidly. But because the possum managed to jump from Australia to New Zealand, it's plausible that it can jump back, and then we get a spillover, right? So we don't want to see this situation. Instead, what we would like to see is sort of a stable situation where the gene drive is present at high frequencies in New Zealand, but somehow remains in low frequency in the non-target population. So the question is, can we actually design these differentially targeting gene drives? So to do that, we sort of extended this basic model to look at two DEMs, right? So now there's two systems of equations that we need to track, one for the frequency of the target population, one for the frequency of the non-target population, and we add this migration element. And then we can study the stable points, or the equilibrium points of this system of equations. So this is an example of such a case. This is a specific gene drive. You can see the parameters up there. And you can see the frequency of the target population on the x-axis, the frequency on the non-target population on the y. And each one of these circles represents an equilibrium point. So there's 9 of them. 5 of them are unstable, but 4 of them are stable, the blue, green, red, and yellow. And the blue and the red represent global loss of the gene drive or global fixation of the gene drives. And these are obviously stable equilibrium. But we also have an interesting one here at the bottom, the yellow one, right? So this is a stable equilibrium where the gene drive frequency in the target population is high and in the non-target population is low. So this is exactly the differential targeting equilibrium that we want. But in this case, I'm just showing you a case with no migration, right? So it's obvious that you can do this thing, like release the gene drive only in the target population. What I'm going to do now is increase the migration, and you can look at the yellow point and see what happens to it as migration goes up. Right? So as you increase migration, you still have this yellow point until you cross a critical threshold, and you no longer see this differential targeting equilibrium, right? So we can see that this differential targeting is possible, but only for low migration rates. But just how low? So we need to study this critical threshold, which we call M star, right? Which is the maximal migration rate for which you still have this ability to do differential targeting. And we can now look at it for all possible gene drives. So this is all the conversion rates and fitness costs for an additive gene drive. And what you can see is that there's a large part of the parameter space that's white, right? And so gene drives in these parameter spaces don't have any differential targeting ability. The gene drive frequencies at equilibrium are always going to be the same in the two populations. But there's a sort of a considerable range here in the middle, the blue-yellow one, where differential targeting is possible. So if we design our gene drive to have these parameters, it's going to be able to do this differential targeting thing. But for most of this range, this is the dark blue. So these are very low critical thresholds. So this is going to be possible only for very low migration rates. And only in this narrow band in the middle, the yellow-white band, you have a gene drive that has differential targeting for fairly high migration rates. So if we want to design our gene drive to do that, we need to design it to fall exactly in this sort of one-dimensional band here. And this is, of course, very, very difficult and probably not practical at all. Okay, so this was the first basic model that we sort of explored to understand what's going to happen with gene drive dynamics. Other people have explored other models. But what we did here is sort of extend this basic modeling approach to look at migration networks, right? So this is a project by a postdoc in my lab and two students, Keith, Jonathan, and Shachar. And what they did is sort of formalize this in the migration matrix form. And what we see here is if we took the same network and the same gene drive and just consider releasing the gene drives in different populations in the network, we can show that, for example, in this case, the gene drive that's released here, this is a differential targeting gene drive. It does what it's supposed to do, spreads only in the target population, but over there it doesn't. So this is the same gene drive, but only in different positions in the network. The same with a gene drive that's supposed to spread through the entire system, right? We just release the same gene drive in two different points in the network, and we get different outcomes, right? So the position of the release of the target population in the entire population network affects the outcome. So in order to see if we can say something about the outcome, right, we're looking at a particular release site, we looked at the properties of the nodes that we released the gene drive into. So these are results of some simulations where we release the gene drive at different nodes. You can see the degree on the bottom, on the x-axis. And what we can see is that if we release the gene drive in nodes with very high degree, the gene drive is going to fail. If we release the gene drive in nodes with low degree, then it's possible that we're going to get fixation. But if we release the gene drive at sort of the intermediate range of the degrees, it's very, very likely that we hit only our target, right? So this local centrality will tell us a lot about the outcome of the entire process. If we look now at global metrics of centrality, we see that they don't tell us as much as the local centrality, right? So closeness centrality tells us a little bit about the outcome, but degree centrality is much more important to understand if you want to know what the final outcome is going to be. The same thing we see when we are looking at the gene drive that's supposed to spread through the entire system, a strong gene drive, right? So the degree centrality, the local degree centrality will allow us to predict what the outcome is going to be, and the global centrality give us less indication of the outcome of the entire process. However, we are not just interested in the final outcome of this process. We also need this process to be fast. And the reason is that the populations are likely to evolve resistance to the gene drive mechanism. So if the gene drive doesn't spread through the entire population and eradicate it fast enough, the population is going to evolve some resistance. So we also need to look at the time to fixation, right? How much time does it take for this entire process to occur? And what we're looking at here is the correlation between the time to fixation and the release site, degree of the release site. You can see that the local degree does tell us something about the time to fixation. So the higher the degree of the release site, the degree centrality, the faster the spread is going to be. And this is because sort of this local centrality gives the gene drive sort of an early boost that spreads to a few more population, and therefore it fixates in the entire system faster. But here, when we look at the global measures for closeness, for example, we see that we get a much better correlation between the centrality of the node, the global centrality of the node, and the time to fixation. So if we really want a fast gene drive, we need to pick one with a high global closeness centrality metric rather than this local one. So this means that we need to think both about, if you want a fast gene drive that will give us the outcome that we want, we need to look at the closeness centrality and the degree centrality of the node and sort of find a sweet spot there. So these are two models that looked at like different aspects of population structure, but there's a lot more to be done. We did some of those models, but of course there are more models to look at. Just show you one result from a model that looks at demography, because we saw that we have a demographic process of crash of the population at the same time as we have spread of the gene drive. And then if we do demography, we also need to think about, soft and hard selection. So that needs to go into the model as well. So Keith extended sort of this simple model, adding demography. So we have another set of equations that describe the population sizes of the two populations, the target and the non-target. And we get this, in some cases, we get outcomes that we saw in the previous model, but we also get some new outcomes, right? We get, for example, we can get oscillations, which we didn't get in the model without demography. And an interesting outcome that I'll tell you a little bit about is this gene swamping and rescue outcome that didn't appear in the model without demography. And what happens here is that if you release the gene drive to the target population, it starts spreading in the target population. It's differential targeting, so it's a bit also in the non-target population. But now the population starts to crash because the gene drive starts to spread in the population. So as the population starts to crash, there are fewer migrants moving out. And the effect of the migrants coming in from the larger population can reverse the direction of the gene drive. So the gene drive frequency can actually now go down because there's more wild types proportionally coming in. And this can be so extreme that it actually generates a gene swamping-like effect, not due to selection, but due to the balance between the non-Mendelian inheritance and gene flow. And the gene drive can disappear from both populations. And then the population can be rescued. So this is an interesting outcome, which may be relevant for sort of a fail-safe mechanism for an experiment, for a field trial of gene drives. So actually, my main point here is that there's a lot to do with incorporating ecology into gene drive models and also to models in general, which we can have a discussion about afterwards. But to facilitate this work, especially in the gene drive modeling community, we developed this platform called Model Archive, which sort of tries to collect all the models in one space and in one sort of coherent platform. And what's nice about this platform is that it's sort of LLM based. So you can just copy paste whatever code you wrote to do your model. And it's trained on actually on TPB and probably some of your papers as well. So the LLM is trained on modeling from our community to be able to correctly generate code that works with this platform. So it works by copying paste code from any language and then generates the model for you in this platform. Right. So we're hoping this will be useful for anyone. I use it for teaching, for example. It's very useful. Welcome. It's web-based. You don't need to download anything. So take a look. The take-home message is that it's important, because we can separate these timescales, the ecological and the evolutionary timescales here, we really need to incorporate ecology and think a lot about what ecology to incorporate and how to incorporate it. And I also want to emphasize that the role of modeling and theory in this field of gene drive modeling is quite unique in the sense that there's a lot of regulators making decisions about, to approve or not to approve gene drives, how to progress with gene drive projects, but there's no data, right? Because we can't, it's too risky. No one released gene drives in the field yet. We only have lab experiments. So the only information that regulators have is what we provide them, the theorists and the modelers. So it's a case where we are directly talking with regulators, and there's no sort of a lot of intermediate gaps there. So again, important contribution, could be an important contribution of our community to an actual technology that's being deployed or could be deployed. I want to thank the collaborators and all the people who worked on these models, and of course, you for listening.